Dear Drug Inspector aspirants, welcome to Plato Online Coaching, and I'm Suchitra, one of your mentor, pursuing my PhD from Usmania University, having a teaching experience of about 10 years. And today, I'm going to discuss with you about TSPSC Drug Inspector Syllabus, its weightage and references. So coming to the Drug Inspector Syllabus, so they have mainly given carbohydrate metabolism. So, under carbohydrate metabolism, there are few subtopics like glycolysis, glucogenesis, glycogenolysis, pentose phosphate pathway, uronic acid pathway, and abnormalities of carbohydrate metabolism. And in this session, I am going to give a small overview about these topics. So, coming to the first topic, glycolysis. So, what is carbohydrates? Carbohydrates are generally their rich sources of energy. Right, and these carbohydrates, after we eat, that will be digested mostly in the mouth by the presence or in the presence of an enzyme, salivary amylase. So after digestion, after entering into the intestine, there it will be entering into the blood circulation. So in the blood circulation, in the blood glucose levels means the normal fasting blood glucose levels are 70 to 100 milligram per deciliters, and this should be maintained. So that will be monitored and stabilized by liver. Hence that liver is known as glucostat monitor. Coming to the first subtopic, that is glycolysis. It is also known as Embden-Meyerhof pathway. So, before entering into the topic, let me introduce two words, that is lysis and genesis. So, lysis is nothing but breakdown and genesis is nothing but formation or synthesis. So, here the first topic is glycolysis, that is glucose, breakdown of the glucose into two molecules of pyruvate and lactate. So, depending upon the presence or absence of the oxygen, it is going to produce pyruvate or lactate. Right. So, in order to produce energy in the form of ATP, that is adenosine triphosphate, which is the energy currency of the cell. And coming to second topic, that is glucogenesis. So, glucogenesis is nothing but synthesis of glucose from non-carbohydrate sources such as lactate, some precursors like lactate, pyruvate, propionate, and glucogenic amino acids and glycerol. So, this mainly happens when during the starvation, when all the stored glycogen has been depleted. And coming to the third topic, that is glycogenesis. So, glycogen synthesis. Right. So, glycogen is going to be synthesized. So, when the blood glucose level increases, so in that conditions, our liver converts the excess glucose into glycogen, and that will be stored in the liver. That is glycogenesis. And next one is glycogenolysis. So this glycogenolysis, nothing but breakdown of the glycogen. So during fasting, this glucose levels in the blood decreases. So in order to maintain the normal level, this glycogen will be broken down to produce glucose molecules for the continuous production of the energy. Next pathway is pentose phosphate pathway. It is also known as hexomonophosphate pathway or hexose monophosphate shunt. And this is an alternative pathway to glycolysis and TCA cycle for the oxidation of glucose. And next one is uronic acid pathway. And this uronic acid pathway is also an alternative oxidation pathway to glucose. So, all the pathways and the reactions and the summary of the reactions we'll be discussing in the next sessions. And coming to the weightage. So, from biochemistry, we can expect at least 5 questions and each question carries 2 marks. So, overall, you will be having 10 marks. This paper will be for 10 marks. Right, and all the questions will be directly only. And this biochemistry, already we have seen, this syllabus is very small. So, this is a small area where there is a scope for scoring almost all the 10 marks. Right, and coming to the reference part, so you can refer the biochemistry book by U. Satyanarayana and U. Chakrapani. And all the B.Pharm graduates' dream job is drug inspector. So I wish you all good luck to become future drug inspectors. Thank you and all the best to future drug inspectors. We'll meet in next session. Thank you.